Hey everyone, welcome back. So now we're going to talk about gene expression and translation. So we just finished talking about transcription, which is the production of the RNA transcript. And now we're going to talk about how we translate it into the functional protein. So in eukaryotic organisms, this occurs in the cytoplasm. So I'm going to write eukaryote. But also in prokaryotes, it occurs in the cytoplasm. So in both cases, translation is always going to occur in the cytoplasm, okay? And translation leads to the production of a protein. So we're going to produce a protein. So protein synthesis is another name for translation. So we use a genetic code which uses the 4 nucleotides. So it uses the 4 nucleotides to code for those amino acids. And for every 3 nucleotides, we have 1 amino acid being coded for. And so 3 nucleotides is termed a codon. So that's a 3 nucleotide unit of the messenger RNA. That's a codon, and that codes for 1 amino acid. There are 64 different codes then that we can produce. And so we get that number by taking the 3 nucleotides that we need for a code and to the 4th power, that's the number of nucleotides that are found in messenger RNA. And so 3 to the 4th power is 64. Another way to look at it is 3 times 3 times 3 times 3. That's also 64, okay? And so there are only 20 amino acids that we have that make up every protein that makes us up, that makes bacteria, that makes plants. We all have the same amino acids, right? But there are 64 codes. So what that means is that the codon table is redundant. Redundant means that there are some amino acids that have multiple codons. And of the 64 codes, 61 of those codes code for amino acids. The other 3 code for the stopping of translation. So they're called stop codons. And so here's a codon table. So when you're looking, and I always like to use AUG just as an example. When you're trying to read a codon table, every single code is in here, but to find the answer or your codon quickly, you split it up into the 3 individual bases. So A is the first base. So we look here and we have to go, okay, so UCA, we know that we're in this section. U is the second base. So we go up here, across the top is the second base. So we say U is right here. So now we know that we're going to be in this row, this 4 mini row section row, and we're going to be in this column. So it's going to be one of these 4 amino acids, okay? And then we go to the G, the last letter. So we go down here, we go down to our group, and we say UCAG. So this is the code that we're looking for. AUG is methionine, and methionine is the start codon, or is the start amino acid, okay? So methionine starts all protein synthesis. So translation has 3 basic steps. We have initiation, which we need energy for. Elongation, which we need energy for. That was my Dudley, sorry. And termination, okay? So initiation starts with AUG, the start codon. Elongation allows for the ribosome to move along that RNA transcript until we hit that terminator sequence. So here we have the ribosome coming together around this messenger RNA. So we have the small and large subunit. Within a ribosome, there are 3 sites. The P site is the site where the first messenger RNA comes in and binds. The first messenger RNA that comes in, or I'm sorry, the first transfer RNA comes in and binds. It's going to be complementary, and it's always going to be at that start codon, which is AUG here. Okay, so in the messenger RNA, this would be A, U, and G. So A, U, G. Okay, which means that the anticodon for this would be U, A, C. And it's going to carry with it methionine. Then another amino acid being carried by another transfer RNA is going to move into this A site. So we had initially this site had the methionine, and then another amino acid comes in here. It was carrying serine. What happens then, the amino acid is going to form a bond with the amino acid here. So like you see this here, you can see in this case it's valine is in this first spot, and it's going to form a bond with asparagine. This bond is known as a peptide bond. And the entire sequence of amino acids is going to move over here to the second spot. And when that happens, this first transfer RNA is now free. So the entire ribosome moves. The first transfer RNA is now in this open spot, and it can leave. So this this E site is kind of the exit site. The P site is the start site, and the A site is just the second site. Okay, I typically don't even talk about the E site, but I wanted to show it since it has a nice picture here. So as soon as valine gets transferred to the asparagine, the entire ribosome moves, and now this asparagine, valine, tryptophan, alanine, serine, methionine sequence is in that P site. And another transfer RNA moves in with another amino acid. That first transfer RNA can leave, and it can go pick up another asparagine. Or I'm sorry, another valine. Sorry, not asparagine, another valine. And then this entire sequence is going to bond to that threonine. And this transfer RNA here can then be released and go pick up another asparagine. Okay. This occurs and keeps occurring until we hit one of the 3 stop codons. So that's that termination sequence. Once we hit that stop codon, the ribosome comes apart. The messenger RNA is then going to be able to be retranscribed, or I'm not transcribed, retranslated, or it's going to be degraded depending on what we need. And the amino acid sequence that we see here, right, is going to be folded into its functional protein form. So let's look at this. This is my depiction of what's going on. So we're looking at, here we have our ribosomal RNA is the yellow. These pink are the transfer RNA, and then this green line is the messenger RNA, okay? And again, I made this for my microbiology class, and so we were talking about prokaryotic translation, but it's the same thing. So here we have the transfer RNA, the first AUG in the sequence. That's the start codon. Methionine comes along, and then we have glycine in the second section with its anticodon there. And so now methionine leaves, or this transfer RNA leaves. Methionine is attached to this glycine, and this is going to keep going. Now we have another one, asparagine. They're both attached. And this keeps going until we hit that stop code. So in this case, UGA is the stop codon. And once we hit that stop codon, the ribosome comes apart. The protein is able to be folded into its functional form. And the transfer RNAs can go and pick up more amino acids. So that is translation. And remember, these are all peptide bonds. And that's why this is called a polypeptide. So there are many peptide bonds in it, okay? So I'm going to stop here. And we are going to get into how we control gene expression in our next video. And so this figure just shows the entire process of gene expression. So you guys can look at this, and I will see you in the next video. Bye.